And so how we started with CRISPR was really when Eugene Koonin visited us at the end of 2005. He was in a PhD defense committee and presented his hypothesis of how this CRISPR system could work. Having several predictions that were there, based on RNA interference and the predicted functions of these proteins. And when he left, and then John van der Roos and I sat together and said, okay, well, we should shift, we should change the topic that we work on in the lab, because we work on something completely different. And we basically started working on CRISPR and chose E. coli to do it. So we were like, okay, let's choose the simplest organism we have available with a CRISPR system, and that is genetically possible to manipulate. And of course, sulfolobus is rather difficult, as I found out during my PhD. That's why we chose E. coli. And and then soon after, a PhD student also started on this topic. So in 2006, we really got going, and we started to investigate this system. And I remember a couple of key moments in this process. So, for example, the moment where the PhD student Matthijs Jore came in with an SES-based gel, showing that these cascade complexes form. Having producing all these Cas proteins and then fishing one out that would co-purify all the other ones at the same time. Suggesting that there was some sort of protein complex. So at the time, we had really no clue what this was doing, but now it makes perfect sense. This is the complex that carries the CRISPR RNA, and that is the essential factor for immunity. I think that was one of the really cool moments. I remember another cool moment where, for example, actually I was on holiday, and Matthijs and Ed Silvestra were there, and they were investigating. Now we were a couple of steps further. We were looking at how these long CRISPR RNAs were being processed by what is now called Cas6, and we had some evidence already from northern blotting that these small RNAs were being formed. So we were looking at the factor that would cut them up in pieces. And then after my holidays, I sat down with Matthijs and Edse, and they were showing me these gels of some sort of cleavage of this RNA, but then in the absence of metal ions, which made no sense. So with EDTA, you couldn't block that activity. So I remember that they somehow were a little bit disappointed, and but I was really excited. Maybe I got re-energized also by the holiday, so I don't know. But that was really the moment where we could start focusing on, okay, what is this band exactly? Can we sequence it now? And which led to basically the CRISPR RNAs and the factors that form them. So I think that was also really an interesting moment. And if I could choose like a third moment, that would be the moment where we found that these cascade complexes can bind to DNA. That was at the time was absolutely not an obvious thing. People were comparing these components to RNA interference, which have, for example, the RISC complex takes a small RNA molecule and will pair it to RNA targets. So that was the obvious one to test. But then once, this was also the work of Magnus Lundgren at the time, finding that these complexes, cascade complexes with the guide, are able to bind double-stranded DNA without using any ATP. So you don't need to add any additional energy into the system. So, and that at the time was really striking to me, that these complexes can do that. So they suggested also that like all DNA in the cell there was some surveillance going on by these complexes, costing no energy at all, only to produce them in order to find their targets. So these were a couple of really interesting moments, I think, in a very rich history that we had, and really enjoyable time as well. So that was a very nice time.